you can also use it to view the data, the metadata, but we are also going to develop a more tabular version of the viewer so that you can have summary and compare different microscopes and that is going to be available for both Omero and also the standalone version. And we hope to integrate it in different websites.
Thank you. That's exciting. Thank you.
Okay. So we can officially start, I guess. And
started recording because I think the stuff and the questions you were answering were interesting. I think maybe we can just make 1 or 2 more minutes because I know a lot of people come in right at the top of the hour. So
people have asked about example Microscope.JSON files. So I'm going to paste it again here. This is a series of different JSON files that we have produced as part of the Nature Methods publication, and you could download them and use them as a starting point for as templates to document your own microscope if any of these is similar to one of your microscopes, but to make it easier. So,
all right, I guess we can get started now. So welcome to the Tools for the Collection and Reporting of Microscopy Metadata workshop. And I'm Claire Brown, and I've been involved in collaboration with this project with Caterina, Joel, and I see Thomas Pengo is here as well. I didn't see Mark, but Mark Sanders has been involved with the project, as well as Alex Regano. So I'm going to have my co-hosts introduce themselves. So Caterina.
Hi, my name is Caterina Strambio de Castillia. I am at UMass Medical School. I am a Chan Zuckerberg imaging scientist, which has allowed me to dedicate a lot of effort to this work about reporting and reproducibility. And I'm very grateful also to the 4D Nucleome project that has allowed me to develop the Micro-Meta app.
Thank you. And Joel?
So hi, my name is Joel Ryan. I'm an imaging scientist working with Claire Brown here at McGill in Montreal. And I guess I was a little bit more involved in MethodsJ2 in this collaboration with, Claire, Caterina, Thomas, and many others. So looking forward to chatting about it more today and getting some of your feedback.
Thank you. So Caterina has really been spearheading the Micro-Meta app part of the application, and then Thomas Pengo and Mark Sanders had developed MethodsJ, and Joel had worked with them to really add a lot of functionality to it, which we brilliantly termed MethodsJ2, and I've been, I guess, involved more in coordination and oversight of the project. So we're really happy to tell you more about what we've been doing today. So we'll just start with a little overview of the workshop. So we thought a big part of this meeting is about getting to know the community and networking. And we'll start with a little icebreaker activity to get to know one another. Then we'll have an introduction to the main project and a general Q&A if there's any sort of questions about the overarching project itself. Caterina will give a live demo of the Micro-Meta app, and then Joel will give a live demo of MethodsJ2. We'll follow that by an open discussion now that you will have seen the two software packages in action. And then we'll wrap up with a bit of comments about future directions of the project. So we'll start out with the icebreaker. So we're going to, let me see how many people we are here now. We're 14, so maybe we'll break up into 3 or 4 breakout rooms. And if you just want to introduce yourself with your institution, your facility, or your lab and your role. And then if you want to share what you hope to gain during the workshop today, and also just to break the ice, if you want to share a fun fact about yourself with your group. So I usually give an example so people don't feel too intimidated. It doesn't have to be anything complex, but I have a nice example. Before the pandemic, I traveled to Australia And when I left Montreal, it was summer. And when I arrived in Australia, it was winter. And then while I was in Australia, it became spring. And then when I came home, it was fall. So I had the whole 4 seasons in 2 weeks. So I always joke that I could put it on my CV that I could do the 4 seasons in 2 weeks. So it was kind of a fun fact. So we'll go ahead and break up into the breakout rooms for about 10-15 minutes. And if you can just try to make sure everybody has a chance to share, that would be great. So I will see if I can manage this. I have one monitor here, so I will stop sharing first. And I'm just going to set them up randomly. So I hope everybody had a good chance to meet some new people and learn some fun facts. I will go back to the PowerPoint. So just to give you a bit of an overview of the project, we really, I think you saw actually we were going to ask how many people had a chance to watch the video ahead of time, or if you had attended the talk this morning, and maybe we can just do it by a show of hands or thumbs up on the reactions. Maybe you can look, Caterina, because I can't see with my sharing on.
Tao, do you need to ask a question, or you're just saying that you watched it?
I think they're just saying they watched it, but
yeah.
So just a few of you, okay. So if you do have time to go back, Caterina gave a really nice 10-minute overview of a lot of information and really outlined well the need for microscopy metadata. I think the fact that you're all at this workshop, we probably don't have to convince you too much of that, but in order to do that, we really have to come to common terms and agreement on what metadata to report. And that's been really great, having these different organizations that are really representing the community. So we have a slide at the end showing all the different groups that have contributed, but Caterina had mentioned earlier, she started this project with the 40 Nucleome, and Bioimaging North America and QuarApp and many other groups have been involved in really spending a lot of time having conversations and just making sure we agree on the terms and agree on what's the essential information that needs to be reported. And for any of us, I think working in a lab or in a facility, you know how difficult it can be to convince people to collect that information. So the workshop today is really presenting 2 tools that our team have developed in order to collect the metadata and do a first pass at the methods section for publication. And the tools were really trying to build on existing resources. So for MethodsJ2, it builds on Fiji. And we'll tell you a little bit more later about another project where we're working on adapting it for Nepari. And then, as Caterina has mentioned earlier as well, the idea is to integrate this with a Miro so that you'll have all of that metadata in your data management system as well. And there's plans to also extend it to include instrument quality control and standards. So we wanted to open it now, just if there's any general questions before we go into the demo of the 2 software platforms. So I'm going to stop share, and you can either use I guess you can use the raise hand feature under reactions if you want to ask a question, or you could type it in the chat. And I know we haven't covered much yet, so if there's no questions at this time, we can go into the demos as well. But
there were questions in the breakout room, so maybe a lot of people already asked, and people are saying, let's go to the demo. Okay.
All right, so I'll pass it off to you then, Caterina.
Yeah, so I am gonna quickly give you an overview of the tool. Before I wanna, so if there are a series of, so after you watch this tutorial, I mean, you take part in this tutorial, there are a series of videos that go through similar things to what I'll tell you again. And so I'm just going to give you a little bit of an orientation about the videos. So there is video 1, which is really an introduction about what information, how to collect, what information to collect, how to collect it, and starting to describe the Micro-Meta app. Then there is a video that tells you about the overall objective of the tutorial. Then what material is needed for you to be able to take advantage of the tutorial. Then there is videos 3 to 7, which actually get into the nitty-gritty of explaining how to do how to do different aspects. And at the end of video 7, there is some description of some more material that you might want to access to learn more. A lot of the so I feel like I've already kind of covered this in the talk this morning, but I just wanted to give you, in case you didn't have a chance to go. So we are only concentrating on the microscopy metadata part of larger set of metadata, which is in overall called we call image metadata. So this is just about describing the hardware of the microscope and the settings that were applied during the acquisition, and also with QUAREB, quality control, and metrics and procedure descriptions. This is, we call it for the NB Naomi or NBO, and now it's also being developed by QUAREB. So we might need to generate a new name, but for now it's called NBO, NBO. And we are basically building everything on a model, which starts from the OMI model and define the instrument with all the different components. And for each component, which literally represent individual hardware components, it defines what kind of fields you need to fill in. And then there is another part that has to do with the image, which collects the settings that were used, that were applied to a specific instrument to acquire a given image. And to do this, we have developed multiple tools. Here you will hear about MethodsJ2, but there is also another tool called Omero MDE. So MethodsJ2, you will hear from Joe, and Micro-Meta App, I will tell you about. And I just want to clarify that again, that there are 2 parts of Micro-Meta App. And the whole thing is based on the OMI model. So first we define the hardware, and in first approximation, the hardware doesn't change every time you take an image. It stays the same, although you could change the configuration of a microscope, like by changing a certain laser if it breaks down, or replacing an objective or whatever. But these will be used, this information will be used over and over again to document all the images that are produced using this specific machine in this specific configuration. And this information is collected in a Microscope.JSON file. And I'll tell you about how to build these Microscope.JSON files, but literally it's going to be a graphical representation of a physical machine. The second part allows you to select a certain microscope, import existing metadata from the header of a given file, and then fill in the blanks of other information that might be missing from the header of the file to produce a setting.json file. And I will tell you about how, a little bit about how to do this. And I want to thank everybody. Alex Rigano is the developer of Micro-Meta App, and you'll hear about Joel, who is the developer of MethodsJ2. So this is, now we can go to the actual live demo. So here I'm going to share my whole screen. Can you see my screen? Yes. Okay. So I hope you have received the link to this protocol.io file. And this protocol.io file contains really instructions and a lot of material, step-by-step instructions that are complementary to the video that you can watch and this live demo. Up here, you can go through the different, like prepare for the tutorial and then get ready to install the app. I hope that I'm not gonna really go through the details of how to download and install the app. We're gonna jump in and actually do the two parts of the app, which is the manage instrument and then the manage settings side of the app. When you first launch the app, whether or not it is, if it is a standalone version, so we have multiple types of implementation. You can download the standalone version, and we are also developing it for Romero. But if you develop the standalone, if you download the standalone version, the first thing you'll be asked to select a home folder. The home folder is basically your repository for your own Microscope.JSON files and Settings.JSON files. So whenever in the app, we're talking about repositories, that's what we're talking about if it's a standalone. It's basically going to be a folder in your Microscope, in your computer, in which you store all the output of Micro-Meta App. So if we continue here, we then actually, something happened here. Yeah, so we have the choice of going into a manage instrument or a manage setting. And the first thing I'll show you is how to manage the instrument. And one thing that is really like an integral part of the model is the ability of deciding what level of documentation you need to use. And in our Nature Methods article on that really describe the community guidelines that inform Micro-Meta App, you can learn more about these different tiers. But the idea is that depending on the type of experiment and the type of instrument you're using and what kind of analysis that you require, for example, you may need to have really minimum information versus very high level complex information. The good news is that you can start by having a low tier and then build upon it, but as you go along, and I'll show you a little bit how to do that. The first part though is I'll show you a relatively complex custom-made microscope. So I'll open it in tier 3, and I'll show you how to import it from file. So if you download it from the website, you can import from file. That means it's not part of your home folder yet. It's from outside. For example, if you download a template from Zenodo or an example file, that's what you would do first, importing from file. Click here and select the file. When this changes a little color, you say continue, and this gets you into the main view. And using zoom in and zoom out controls, you can zoom out or you can zoom in to focus on closer on the different parts. But one thing that I really want to make sure that you understand is that there is a lot of information here. So we do have lots of possible. So if you open a Microscope.JSON file from the first time, you may want to just scroll down to see if you can see everything. And in order for you to see more, you can also minimize this part here. So we capture, so you see, I told you about hardware, but here I show you software. So we capture basically the unchangeable parts of the stuff that exists in associated with your machine. That is a priori before you acquire any images. So the first thing I can show you is about the general, what's written here. Here you can see general information about the microscope, and then to access that information, we can click on this button here. And here you can see the different like the general structure. So we have tabs, and then sometimes we have internal tabs and maybe internal subtabs. And in each case, you'll see that there is always a general tab, and then there will be other types. For example, in the instrument microscope stand, we have a general, manufacturer spec, and the specific inverted microscope stand tab. Each one of these, you will see that the general is always the same for all components. The manufacturer spec is always the same for all components, but the third tab is specific for each component. So be sure to navigate through all of the different parts. And again, I just wanna stress again that these fields are created by Micro-Meta App on the basis of the model. So if the model changes, which we are planning to change it and continually evolve it, especially now with the manufacturers, we're working on this on the camera side, but we're planning to do it on all the other components. These fields might be updated, but the Micro-Meta App will allow you to be always in sync with what is the current version of the model. As you can see, another thing I want to show you is these green dots. What are all these green dots? These green dots tells you that the information associated with each one of these components is validated, and it's validated and tells you here it's validated at tier 1. And you can also check if it's validated at higher tiers. So if you open a microscope at tier 3, like what we did here, it will contain all the fields for tier 3. But if it is validated at tier 1, it will allow you to validate it at a lower tier. So this is a very good trick to start compiling information. Initially, you want to start low, start filling in the information that you know about, and then add more information as you gather it, as you find it, and make sure that it gets validated at a higher level if you need to, depending on your microscopes, on your experiments. So in this case, this guy should be able to validate at tier 3, and you can see that all the dots remain green. So if I open, for example, an objective, now you can see that, again, there is a general objective tab here. There is a general, contains like a custom name for this objective, a description. Then the manufacturer specs contain the information about who produced this objective. And then the objective fields are about characteristic that you need for describing specific the objective itself. Okay, so I think I have to move on to create the next part. I will show you how to create from scratch a microscope. So that let's say that so the idea is that if you download an example file from the Zenodo repository that I put on, that I can again share as soon as I'm done in the chat, and you find something that is similar to what you want to document, you should start from there. And then you should save it as a new microscope. So save it as new. It will create a new ID for that microscope, and you'll be able to get rid of all the stuff that you don't need. And actually, in the latest version of Micro-Meta App, I want to show you that there is also a copy feature that allows you to take an existing element, copy it, and then you can actually paste it. And then you don't need to refill everything. You can just take everything that is in common and basically just fill in the things that change. So this should make it easier for you to do your documentation tasks. But if you don't have, if you want to start a documentation completely from scratch, then I'll show you how to start from scratch in the next step. If there are any questions up to now, please let me know.
Caterina, there was one question of whether you can have stereo microscopes.
So we don't have that specifically implemented in the model yet. The next extension that we are implementing is for confocal microscopes, both laser scanning and spinning disk. So we can definitely consider that. But if there are, everything that is in common with the wide field, it can be used to start at least documenting the stuff that is in common with the wide field. But otherwise, we have to evaluate that. Yeah, so that Judith also answered. Okay, so in order to start from scratch, here I have in this protocol IO, I have, so here actually tells you that if you wanna review what I just told you, there is a video that tells you pretty much everything, you know, a little more of what I've told you so far. But let's say that you wanna start from scratch. Here on protocol IO, you can find, for example, like a different, the link to different components that you may wanna record, but you can also do it with your own microscope and just get started with it. So here I'm just gonna start with tier 1, and I'm gonna start with create and inverted from scratch. And it's gonna be a completely new microscope. And so there are different ways of proceeding, but what I like to do is I like to give a name to this microscope. This should be a name that you know that in the facility, this microscope. And then the good thing that you should know is that if you hit confirm, it tells you what fields are missing for that specific. And I'm just gonna enter them, at random right now for that specific tier level. So now I'll be able to confirm it. And here, this part here becomes green, and I can save it as a microscope. And this is now going to be part of my repository. So it's going to be in the home folder of Micro-Meta App. Or if you're running it in Homero, it will be part of the Homero repository for Micro-Meta App, as soon as that's going to be available, should be soon. I want to document a very simple thing. So let's see, what do I need? I need a light source. So I'll put a laser here, and then I'll put an objective, here, and then I'll put, this is a simple filter cube with an emission filter, an excitation filter, and a dichroic. Sometimes this thing gets stuck. Okay, and then I'll put a detector. In particular, I'll put the CCD camera or CCD camera. Yeah. So now this is all of these new items are not validated at tier 1, but I can already save it. And if I need to open it again, what I do is I will now find it in my home folder under the C manufacturer, and I can open it and continue my work. And in order, for example, to start entering information, we can go through the process. I mean, the way I like to do it is, so this is the link from the protocol IO, and there is information about this objective. And this would be great to have as automatic as possible, but if you have to do it, you can cut and paste information. And for example, here, I can say that this is, this is the model. It is a size objective, and I have the item number here, and I can cut and paste it and enter all the different information. And I can just say that this is the objective. I think it's a 63X for scratch, which is this specific microscope. And then I can enter all the different information following the information that is described here. Sorry, I have somebody calling me. And so once you have entered all the information, you can then validate, and that's it. Sorry. and you're now validated the objective. And then you save it just because you want to save your work. You can do the similar things for the other elements. In the video in particular, I explain in more detail about how to set up a filter cube with all the different filter parts. I don't have time to do it now, but I want to show you what you would do to, if you want to now, like you have a different experiment you want to do with this same microscope, and you want to now increase to validation level 2. And so I can open again this microscope, and now it will allow me to move to validation to 2. And, for example, the objective will not be validated because there will be new fields that I need to fill in for bringing it to validation level, to tier 2. And here, for example, I have more information. I have the working distance, and I can put it in, even though the first time I didn't record that information. I can now do it. And I can see that this is doesn't have a correction color. And I think that's it. And that's all the next information I need for tier 2. And now it's validated also at tier 2. And of course, I always want to save. Any questions up to this point? Okay. Now we're going to go through the manage settings. And in the manage settings, I'm going to first show you the existing files that you can download from Zenodo. So what happens is here, the logic is that we first have to select the microscope, then we have to select the image, and then we start creating a setting file. So the first thing here, I can show you, for example, I can load it from, in this case, I actually don't need to, I can load it again from the download. Oh my goodness. My mother is trying to get me and she won't give up. That's what mothers are like. So when you go to the next step, you'll see that this thing advances to the next step, which is to look for the image that you want to document. All of these you can find and download from Zenodo. It will take a little bit for bioformats to process the content in there. Then you go to the next step. And in this case, I'm actually going to use an existing settings file that I have created. The way that I like to call settings file is like I use the name of the image, plus I add acquisition setting at the end, but that is not a mandatory way of doing it. You can find your own convention. And then we are now in the main window of the settings. And I want to show you what is the logic here. The logic here, for example, let's look at settings. The logic is that this file knows all of the different objectives present in this microscope. And you can, in this particular case, this image was acquired using the 60x plan apple. But if I would start from scratch, I could choose this and edit. And then I could then enter additional information, for example, about the immersion liquid. And in this case, actually, some of this information can be imported directly from the header of the file. So another example is you select, for example, the sample positioning devices that you used. So you have the list of all the devices and you choose which one was used for the specific acquisition. Under general settings, you have access to the information, for example, about the pixel structure that was imported from bioformats, but you also have, for example, the ability of selecting, for example, what stage insert was used, what focal stabilization device was used, what acquisition software and what modules were used for the acquisition. I will spend a little bit more time telling you about the channels because, in this part here is quite straightforward. What you do is you select the different components you used for that specific image. In the channel, what you can do is you can actually, again, bioformats will tell you which channels are present and expose what are available. But then here you can do a little bit more. You can, this is basically the schematic of a light path, and you can enter the different components that are, again, available there. For example, here I've already selected a specific laser, but if, let's say I didn't have any, I would have all the different light sources available, and I could choose the one that I want to use, that was used, and I can then enter settings that were applied to that specific one to make it so that it gets validated, and then it becomes, the dot becomes green. Same thing can happens with the camera, with the different parts of the light path, such as, for example, the excitation filter, the dichroic, and the emission filter. Importantly here, in these little buttons, I can hide a lot of complexity about actually what are the different parts of the light path that were used, and I can put them in a specific order. As long as those are recorded properly in the Microscope.JSON file, they're all available here, and I can put them in a specific order to add complexity and detail to the light path, which is something that, for example, is not available on the header of the file. And under channel settings, I have more information about, for example, the fluorophore and other informations that could be associated with the channel. Do I have questions that I need to address before if I do until the last part?
I don't know if we need to address it now, but there was some questions just about having templates, and so I reposted the link for the example Microscope.JSON files, but just sort of the general comments of whether there could be a library of Zeiss objectives or a library of standard configurations of microscopes from the different companies.
Yeah, so that's what we really want to do. So as part, so we are actively looking for additional funding to do a lot of this. With Claire, we are working to get CZI funding to do some of this. I'm also working, hopefully, to get some NSF funding with the core marketplace people and the RRID people to be able to build repositories of microscope configurations. And then we are working with the manufacturers to make sure that, not immediately because that takes time, but that all of this will be made automatic. For example, with the camera manufacturers, the idea is, as I said, we are working on revising the model so that when you buy a camera, you will be given a JSON file that you will be able, for example, if you want to still build a configuration like this, graphical configuration, maybe it's useful for your users, then you will be able to drag a camera that is already pre-filled. You don't have to do anything. You don't have to enter any information. You'll just enter it. You'll just drag it in and place it, and it's going to be all ready to go. So the first step toward that is we did already implement the idea of cloning. So if you have more than one filter of the same manufacturer, you can clone it and then just change the individual characteristic of individual filters instead of having to re-enter everything manually. But we are doing this, and the idea is that we want to implement the idea of accessing pre-filled components that are shared by manufacturers. One thing that would be great to do and could be done relatively quickly is, for example, with filters and objectives, because those are, there is a lot of information is already available in the public domain. For example, Zeiss exposes a lot of this information. So, for example, if we could do that, so that these could be made available in a way that we can use it directly, would be great. So we are working on multiple fronts to achieve that goal.
I would just add, if you're talking to your microscope manufacturers, tell them, I went to this workshop, and oh, wouldn't it be great if we could get a JSON file from Nikon? And I know from working with companies for a long time that the more they hear a request, the more likely it's going to make it up the chain of, if they hear it from a lot of different places about the same request, then it's going to have more likelihood of being implemented. There's another question.
Oh, sorry.
There's another question here about turf microscopes, if those can be implemented.
Yeah, so we definitely have part of the model. There is already a model for turf. It's not implemented yet in Micro-Meta App, but that's definitely up one of the next things we're gonna do.
And one last question, Michael was asking if you've talked to Thor Labs and whether they might be more likely to buy in earlier than some of the other microscope manufacturers.
Thor Labs is one of the companies we're talking to, and definitely we see that smaller companies are definitely very interested. So yes, it is smaller companies, and Thor Lab is definitely, interested in already. I mean, a lot of what they do is along this line. So yes, we are working with multiple different, and Thor Labs, I'm pretty sure, is one of the people we are talking to. Okay, so do I have a couple more minutes to show how to create a new? Okay, so here in this case, I showed you, what's going on here? An existing setting file, but let's say that I realized that the transmitted light channel is not present, so I can create a new channel here and then select it and edit it. And this is what it looks like when you start from zero. So what you do is you select, for example, the transmitted light. And in this case, there is only one camera, so you select that. And then you may want to select other pieces here, which I'm not going to do now, and put them in a specific order. And then you want to save, like you can say that this is transmitted light. And then you have different choices here that actually come from a specific ontology, so that you can find, let's see, bright field, okay, and then you can say that this is the transmission light path, and then enter. Obviously there is no fluorophore in this case. And it's bright field, and here I'm gonna use, and then it basically allows you to at least start confirming the presence of this channel and validate it, and then you can enter information about, for example, the power that was used. I'm just gonna enter random numbers here, and the different characteristic about the settings that was used. And I'm just gonna enter numbers here.
Sorry, Caterina, is it correct that so every user will need to do this with every experiment? Because I mean, some of these things are constant, like your transmitted light settings, but your gain and whether you have 16-bit or 14-bit may be different from experiment to experiment. So I'm just confused at what state of the process this is done. Is this the person configuring the system, or is it the actual experimenter who needs to be doing this?
So this is obviously, it's a real important question. So as far as defining the hardware, obviously it can be done once and that's it. As far as defining the settings, we will need to do a better job at automatizing some of these aspects. For example, one thing that we're working on is you enter it once for an image in a data set, and then it can be applied to multiple images in that data set. But at some level, the idea is that we want to be able to capture this information for the data. So it is, so like it doesn't take very long to enter this information for one image. It would be really terrible if you would have to repeat it for each image in a data set. So if we can make sure that that is, you know, propagated across the data set.
I would even argue that, like in the example you're doing now, most of this is boilerplate. There's just one or two things that may be different, and so it would be very important to have, you know, the actions of the users only be those one or two things, and the UI focused on those one or two things and not everything, because people will not do it. It's not going to happen.
Yeah, no, I mean, of course, we are going to have to do a, so for example, one thing that can be done on that front is, as I said, propagate across a data set. And the other aspect is, having templates. So if you do it once, you don't have to do it again. You can reuse the settings file for the next one, and you just change the things that are relevant to change. A little bit more. So, I mean, for example, we definitely would love to have input from users that start using it. Like, how do we choose which one has been changed across individual images? And, you know, the other thing is to make sure that these things are captured in the header of the file, so that when we import it, all of it is, more and more of it is going to be imported automatically. But we have to go through these stages of generating the model and start providing tools to at least start collecting this information somehow, and then work with the manufacturer, work with the users to get to a point in which it's going to be much more streamlined. People are not going to do it. I totally agree. Unless there is reasons, and, you know, but if we find a way of meeting people somewhere along the usability spectrum that is closer to them, while we're waiting for things that are done automatically by the manufacturers, and we're working with the manufacturer toward that end. So we're not just doing this, we're doing all sorts of things to get to the point in which all of this information is going to be automatically provided. But in the meantime, what do we do? That's the point.
Yeah, no, here in this specific case, my inclination would be to, at the very least, have these channels predefined for people.
The channels are gonna come in automatically from the bioformats file. So in this case, I just showed you an example that is not really a great use case, because it would come in, it does come in from the bioformats, and as much as possible that we can find in bioformats is pre-filled. But unfortunately, not everything is. And as you know, different manufacturers provide different subsets of the fields.
Yeah, and they have different names and.
Yeah, but we are using bioformats to map that as much as, you know, because they've done a great job at helping us map it. So we're not starting from scratch. And then, you know, Joel now will tell you about MethodsJ2, which will help you do some of this stuff in a slightly different way, and also generate the methods section that are written out. So, you know, we need to be able to make it as easy as possible, as automatic as possible, and at the same time also provide user value at the end. You know, for example, everybody has to write the method sections for the paper eventually. So if we can maybe do a little bit of work here, minimize what they have to do, but then the big carrot is that once you have to write the paper, a lot of it is going to be done for you. So that's the idea, but obviously it's a big work in progress. Okay, I think unless there are other questions, I can stop sharing.
Yeah, I think we're good.
Well, I can stop sharing anyway, even if there are questions.
I think we've covered everything in the chat, unless there's something else someone has before we go on to MethodsJ2? We'll have time for more discussion after that as well. So I think you can go ahead, Joel, if you want.
All right, let's see. Okay, can people see a slide? All right. So hi, so following Caterina's demo on Micro-Meta App, I'll tell you a little bit about MethodsJ2, which we kind of built sort of piggyback on these JSON files, these hardware JSON files that are generated in Micro-Meta App. So I'll be talking about MethodsJ2. And so just very, very briefly, I'll go through a couple of slides. And the idea of MethodsJ2 is really more on the microscopy methods reporting side. So really the materials and methods text that people put in their papers and their publications. And without going too much into detail, well, complete method sections are very, very important. This is reporting the actual experiments that were done on the microscopes. And very simply, you can think of many advantages to writing a complete and well-detailed method section, such as, well, it helps evaluate the work as much as for the reviewers, as for readers. You will get a better idea of how the weight of the conclusions of an experiment if you know how the experiment was carried out. It enables reproducibility, so other people want to repeat or build on work that has been done. It enables data reusability, so well-reported and well-annotated data. It enables other labs to use it, so something that's a little bit more common in the sequencing world, as I discussed with Nico earlier in this workshop. And slowly but surely, with some of this work that we're doing, it will slowly become kind of required by publishers to put in complete and good method sections. And this is all based on some of the work that Thomas and his collaborators did in the past, where they found that most papers, do not offer enough information to replicate an experiment. So you kind of have to, make it from scratch, so to say. So to help with this, building on Micro-Meta App and MethodsJ, the original version, we made MethodsJ2 in order to sort of have a little tool that runs in Fiji to help people write their method sections for their papers, their reports, thesis, kind of wherever you need to sort of report these experiments in detail. Just before jumping into the demo, we also have a little bit of a playlist, so sort of giving an overview and the required information, as well as a demo itself. In terms of required files, this runs entirely in Fiji, and I'll say Fiji, not ImageJ, but Fiji, because we're really reliant on bioformats and a few other goodies that are standard in Fiji and not necessarily standard in other builds or distributions of ImageJ. And then a few files that are available on the GitHub. The MethodsJ2 Python script. It is a Python script written specifically for ImageJ, so it will not run well at all in a Jupyter notebook or a regular Python IDE. So this is available on the GitHub. And then it requires an image file. So again, we have a demo on the GitHub, which I'll open in a bit. And of course, it requires a Micro-Meta App hardware specifications file. So the idea is that hopefully the core facility or the Microscope custodian built up the hardware specifications file and then distributes it to their users in the facility such that they can, essentially build their method sections and report their work. So these three files for the demo are available on GitHub, and of course Fiji is of course all open source. As a general workflow, we're going to load Fiji. We're going to load the MethodsJ2 Python script and run it. Within the script, it's going to ask us to load first an image. Then from that image, the script is going to try to extract a bunch of metadata from the header. And we're still working on building that up a little bit more than what it is right now. And then it'll ask us for a Micro-Meta App hardware specifications file, which will then allow us also to source some information from there. Once that's going, we're going to go through a whole series of dialog boxes that are going to be preloaded with the information that's extracted from the image and from the hardware file. So hopefully the information that's found will be there, and essentially we go through a step of validation, essentially. And the little bits of information that the software can't find just yet, we can kind of fill it in. Very often with these sort of drop-down boxes that will be populated with hardware that's available on the system that we've selected. And if all that works out, then at the end, we end up with a little bit of a text blurb that is much longer than this, but something that can be copy pasted into a methods section, such as, well, images were acquired on an Axio Observer Z1. We realized that we don't actually have an Axio Observer Z1, which Claire had pointed out in the past. Anyways, inverted microscope and so forth. So something that we can copy paste with hopefully minimal grammatical things to change. So I'll stop the slides and we can jump into an example real quick. If there are any questions, of course, before we get going, I'm more than happy to take them. And I'll just try to see where all these files are. Okay. So in MethodsJ, this runs like any macro or script. So you can access a script editor or a macro editor simply with plugins, new and macro. And that should open one of these windows here. And then we can simply find the Python file either with file open in this macro editor window, or we can also just drag and drop Python file directly into the main Fiji window. And so with that, we have the script that's loaded here. So it's a pretty long and convoluted script. It requires internet access as well due to a structure file that's online. And if we're all set, then we can simply run it. And it'll take a couple of seconds to instantiate. We get our first serve window here, which essentially asks us to load an image. So ideally a raw Microscope image file, hopefully with a decent header to it. So I have a 3-color image here that we acquired on one of our Zeiss wide fields. You can hit the browse button to go through a folder selection or just drag and drop an icon directly in here. We have a couple of options as to whether or not we want to display the metadata. So both the original metadata for that Fiji kind of sources, as well as the bioformats OME XML metadata. And it's handy to have these open because sometimes they contain information to kind of prompt the user towards one option or another. And there's an output that's saved. Essentially everything that we put into the system, it'll be saved as a CSV file, and we just select if we want to save it in the same folder or if we want to select a different folder. So again, this would load a folder selection window. For now, we'll just do the simplest thing and save it in the folder. So I'll click OK, and we're going to get a bunch of windows that are going to pop up. And that's a little bit scary at first, but we'll go through them. And so first, on my other screen here, the image loaded, which is kind of the most helpful. So we have here this 3-channel image. So we have DAPI, phalloidin, and mitochondria here. And then we have a few windows that open. So first is this log. So this is the Fiji log that just prints out everything that's being done. This does contain valuable information, but it's also saved at the end in our CSV file. So we don't really need to pay attention to it. It does become handy if something crashes, because then you can see up to where the script kind of went. For now, I'll just move it off screen. And we have 2 other windows that loaded up. So one is the metadata that is directly from the file header. So this is a CZI file. It's a Zeiss file. So it's in this sort of Zeiss format with those vertical slashes between all the tree settings. And we also have the OME metadata. So that's that which has been sourced essentially by bioformats, essentially in the XML tree structure. And so these are handy just to prompt the user if anything's there. We don't kind of access it directly, but it's always kind of handy to see. Finally, we have the first of many windows, and the first one doesn't really have anything to do with the hardware or software metadata source there. This is really to prompt the user to put in information about the experiment. It is not required to run the software, but it is information that's good for a method section. So in sample description, I'll put BPAE cells, so bovine pulmonary artery cells. Sample preparation could be, stained with DAPI, phalloidin, AF488, and Mito Tracker Orange. And you can imagine this is all in Fiji, so it is customizable. If your lab always uses the same, a subset of samples or fluorophores, you could have a drop-down menu instead of just an open string field. The bounding medium is vector shield, not vector field. And cover glass is 1.5 glass cover slip. And the sample folder is simply a glass slide. Again, this is just more to provide information. This particular information here is not formed into text simply because there's such a variety of samples and preparation methods. It's hard to find standard bits of text that fit properly. But at least the information is recorded somewhere, and you know, it will serve essentially to prompt the users as much as possible. So we'll say OK. And here we start getting some of the information that's been sourced from the metadata. So just simple things like image dimensions. So we have the image width in pixels, the image height in pixels, the number of slices, the number of channels. So this was accurate. We have 3 channels and only 1 slice, 1 Z-plane and 1 frame. The dimensional order of acquisition, which is also very useful to kind of determine how the experiment was done. The pixel size, and then the voxel size, the depth here, and the time intervals. There are none particularly in this case, so we have NA, but hopefully those would be sourced by the, well, leveraging bioformats. If there is a mistake, the user can go in and change these things, so just add numbers, but we're hoping that it gets it right. If it gets it wrong, then it's up to the user to kind of fill in the appropriate data. So here, everything is right, so we can just kind of move forward. We'll say OK. And then we're being prompted to open to load a Micro-Meta App hardware JSON file. So here we have one for the wide field that was used to acquire this image. So I'll just grab it from here, drag and drop. It's ABIF axiovert1.json. And of course, you can also use the browse button to go through the folders and find it. And we'll say OK. And then the next menus that kind of that are kind of put together are mostly sourced from this Micro-Meta App file. So first we choose a descriptor for the system, so wide field epifluorescence. We could also choose other types of configurations. This is really just for the text right now, but the idea slowly is to link this to hardware kind of as we go. And acquisition software, this was sourced from the hardware JSON file. The drop-down menu only puts in the name that was put in Micro-Meta App, so I just called it Zen because most users will know it as Zen. But behind the scenes, the software will go get the version, or sorry, the script will go get the version, the manufacturer, and all those other details that we feel are necessary for methods reporting. Since there's only one software, then there's only one element, this drop-down menu. So pretty simple, very simple. So we'll say OK. So again, this is not so difficult. And here we have our objective information. So the metadata, the image metadata suggests a 63x with an NA 1.4. It takes that information and tries to find in the Microscope metadata file which objective would be the closest. So it found a 63x NA 1.4 from this selection of objectives here. So there's on this system, there's a 10x, 20x, 40x, 63x, and 100x. So I've managed to do the text matching essentially to figure it out based on the magnification in the NA. So if you have multiple objectives with the same magnification and the same NA on the same system, it might get it wrong, but at least it preloads this. So again, this is just a validation step. We don't have to select anything, so we can move forward, say OK. And here's where we put details for all the channels. So the first channel here, as you can see here, is DAPI. So I'll just give it a name. I'll call it DAPI. We choose the light source, the excitation filter, beam splitter, and emission filter, as well as the detector. Detector is the easiest because there's only one. The light source intensity, it didn't manage to find it here. I believe it was 20%. But we have the excitation filters. This is just the first in the list, so it kind of works out. So the DAPI filters for each. We'll say OK. And then it asks some information about the camera itself. So the exposure time, which was found in the metadata, 100 milliseconds. The gain, if it's anything adjustable or you have to work with, and the camera binning. So this was a 2x2 binning camera, so we can select other things if needed, but it managed to find it. So we'll say OK. And now we move to channel 2. You'll notice that the image also changed the channel, which is a helpful reminder. And this is phalloidin, Alexa or AF 488. And same thing, we'll change the light source intensity. This is 25%. We'll have to select the actual filters that were used here. So the excitation filter is the GFP excitation filter. We'll use the GFP beam splitter as a dichroic and the GFP emission filter as an emission filter. We'll say OK. And so we're going through this information for each channel. Again, the exposure time, gain, and binning. These are found automatically. If they're not found, there'll be nothing, and the user has to input it and hopefully has to sort of decide whether or not the metadata header is sufficient in the original metadata file. In this case, it's pretty good. So OK. And finally, we have channel number 3. So I'll just put mito tracker orange, and the intensity here was 30%. And here we'll just select the the filters again. So in this case, orange, we use the TRITC filter set. So that means a TRITC excitation filter, a TRITC beam splitter, and a TRITC emission filter. The detector, we only have one camera on this, so that's the AxioCam 506. And same thing, exposure time found here at 200 milliseconds, and camera bending is fine as well on there. So we have all we have most of the channel information that we asked the user to validate. The last step is optional devices, or what we decided to call optional devices. So these are things like stage incubators, environmental conditions, which you may very well have on the microscope, but you might not use. So you'll almost certainly use an objective, but maybe the stage top incubator you only use for live cells. These cells were fixed, so I didn't actually use it. Same thing for a focus stabilization device. You might use the definite focus or perfect focus or what have you for longer term experiments, but you might not use it for a given run. So we call these optional. For today, let's pretend that we used the incubator. So I'll just check this. I'll say OK. And then here we get a last sort of menu here where, well, we use the LCI stage top incubator at 37 degrees and 5% CO2. So I'll put this in. And then at the end, this is something for core facility people. We added a window here to sort of encourage people to acknowledge core facilities in their papers. So please remember to acknowledge the imaging core facility and staff in your publications, since a lot of imaging facilities do rely on this. And again, this is all custom. Would be relatively easy to customize. So you could have this pre-filled with your core facility, or put a drop-down menu if it's one of something like that. Same thing for staff. So name of a staff. And research resource ID, so an RRID. So in this case, I'll just put the advanced imaging facility at McGill, and I already have a typo. And if you got help from a staff member, you can put their name in, and then a resource ID. So research resources IDs give a specific code that will link to the facility. So we have one here. Basically, if you Google this code, you'll find the facility. So it's kind of a better, good identification number rather than the various acronyms and names that different imaging facilities have. And finally, we're done. We click OK. And we get this blurb here in this Java output, which was a late addition from Thomas, I think, which was very, very helpful. And this is pre-selected and I believe pre-copied to the clipboard. So I'll just say copy. We can load our Word file here, so where we would have some sort of manuscript on the go, and we can just paste it. And we get our something pretty close to a decent methods section text here, where that's it. So we have a little warning sign here that it was generated automatically. So please verify for accuracy and grammatical correctness. And I won't read the whole thing, but you get something along the lines of images were acquired on an Axio observer, compound, commercial, custom, modified, inverted microscope from Zeiss, configured for wide field of fluorescence microscopy, controlled with Zen software, version 2.6 Blue Edition from Zeiss, equipped with a 63x NA 1.4 apochromat DIC oil objective, also from Zeiss. So a lot of the sort of background information in the hardware JSON file can then be added in. The image dimensions, and then we have these sentences for each channel. So they're kind of a repeated structure. So users can decide if they want to make it less robotic if they want to. But DAPI was excited with an Excite 120 LED light source set to 20%. Wavelength selection was carried out, and so forth. And then the next channel is a similar structure. Phalloid and AF 488 was excited with the Excite, and so forth. And so our three channels are there, including the cameras, the exposure times, binning, etc. And finally, at the end, we have one of our optional things here. So environmental control was maintained with an LCI stage top incubator. From LCI, cells were maintained at 37 degrees and 5% CO2. So it's really just stitching together little bits of text. So there is some work to be done in making it a little bit more either grammatical or more syntactically pleasing. And finally, we have a little acknowledgement section here. So just a little thing that we can copy paste, hopefully in the acknowledgement section. So images were collected and/or image processing for the analysis was done at the advanced bioimaging facility with the assistance of me and our research resource ID. And so this just kind of helps and prompts people, reminds people to acknowledge imaging facilities in their work. So that's most of it. The other output, in addition to, I see the comment here with regards to gain, and yes, I thought I had that solved already, but I will update that. There is a mistake here with regards to the gain, with gain set to, and yes. So since I didn't put any gain, we have that little bit of text. That would be something that I thought I had solved already to remove there was no gain, but we can go in and fix that, and something that is also customizable in that sort of sense. So yeah. The other output that I do want to mention, so of course this is simply copied to the clipboard. In addition to the log file, it also generates a CSV file. So that's this file here that was just generated now. If I open this in Excel, we'll give it a moment. It's very simple right now, but we wanted to have some sort of tabular output with some of the information essentially that was put in, and sort of a bit of a reference to the information from the Micro-Meta App file. So simply 4 columns, essentially the label. So what was the thing that was requested from MethodsJ2? So things like, well, number of slices or image width or something like that. So really each one of either the units in each one of those dialog boxes, be it a drop-down menu or a string field or something like that. What was the metadata value that was found by using bioformats, essentially? So this is here. And what was the user input value? So that can either be what was manually typed in or what was validated, so what was left by the user. So for example, we did not change the image dimensions. So the image metadata value is the same as the user input value, quite simply. And finally, the 4th column is the Micro-Meta App schema ID. So for a lot of this equipment, it refers to a specific component in the 40M model. So for example, the objective, if you're looking for the objective in the hardware JSON file, it's called objective with a capital O.JSON. And so this can relate to, this name here can relate to this object in the JSON file. There's still some work there to make it a little bit more precise, because this is actually the name of within a dict that contains this objective.json, but it at least refers to the specific hardware, because, well, it's a huge file and can be confusing to kind of find what is what. And at the end, also the text generation appears, as well as let's see, the version that was used, the time at which it was run, the image that was used to generate this methods text, as well as a few other a little bit more information there. And so that's it. That's sort of kind of MethodsJ2. Of course, we're always kind of working on and finding these little bugs, especially with the text stitching. But as we get more feedback, we'll be able to kind of adapt it. Perhaps one last thing to point out, if I have just another moment or two, is it runs essentially on this script. This script is very kind of actuating, so it's kind of doing things like creating those dialog boxes and whatnot. But a lot of the information that's used to generate the dialog boxes is in yet another JSON file, which is online. And this is something that looks a little bit daunting at first, but is a little bit more customizable. So every one of the dialog boxes that we had is essentially is a little bit generic. So the script, the MethodsJ2 script, just says, make a dialog box, and how to make it is going to be with these elements here. So if I just scroll up to something that maybe makes a little bit more sense, or that we saw already, I'll try to find the objective one perhaps that makes most sense. So this is an example of an instance of this. Essentially, the dialog type is add choice. So for the objectives, we had a drop-down menu that's add choice. And then we have it say something like the title will be objective. That's known as the dialog box. The setting, so what it'll say just in front of the drop-down menu will be select objective : space. What is the information we want it to go get from the Micro-Meta App file? So we want the magnification, we want the NA, correction, contrast modulation. These are all part of the model, essentially. So what is the information that we want to go source? And then what is the actual blurb here that we add in? So equipped with a magnification, NA, correction, and so forth, objective, and finally in brackets at the end, the manufacturer. So this is also something that's customizable if you have a microscope with either very specific things that you're using over and over, either samples or things like that. You could relatively easily build your own dialog boxes that would be kind of generically built up like that. Or if there's anything you want to change, especially with syntax or something like that, if you don't like the way these sentences are constructed, you can easily change it relatively easily change it to something else. Right, so I'll stop there. And we'll did I miss a few questions? And if there are any questions, people want to unmute and chat, that's also fine.
So maybe if there's any questions specifically about MethodsJ2, we could start with that. There was a question about the environmental control in the CZI files.
That's true, yeah. So you're absolutely right. That'll depend on the incubator. So to be fully transparent, I haven't coded that in yet for MethodsJ2 to go get the information about CO2 from the image header. In some cases, though, that will not be there. So, for example, our incubator is a separate unit that's not controlled by Zen, and so we turn it on and off manually. So that's really up to the user to remember. So in this case, it has to be put in manually. It would be, yeah, but that would be something to add. Again, leveraging bioformats to go get the information from the CZI file, and I guess have like a function that if it detects it, it puts it in. If it not, well, it has a string field and reminds you to add it. But yeah. And then,
any other questions on MethodsJ2? Okay. So our plan right now is just to open it up for a more general discussion, to just, I guess, get feedback from the community on what you've seen. What do you think of what you've seen? Are there, what's on your wish list, I guess, for... We've heard about a few things, turf and confocal and so on, but we'd really like to have, open the conversation and hear from you what you think and where you would like to see this go.
Yeah, first of all, I want to, applaud everyone working on this. This is it's great work. It's very needed. It's always difficult to get people actually together, and this is a very brave and great attempt to do so and standardize somewhat as much as possible. Really awesome. I'm just sitting in it now. I'm actually right now we're dealing with similar problems in the place I work. What I see as just looking at it now is just the amount of work involved to set it up. On the one hand, I very much want it. Do I want to put all the work into it and have all my users put all the work on it? I don't know if it's going to happen. So that's kind of and so, you know that message as well. Like, it is as much as can be, and I totally understand. It's and the chicken or the egg. You need to get it rolling before it becomes easier. So I don't know what advice to give there. It's like, keep on going. I will try to do it. Put the work in.
If I can.
One thought I had, is also, I love Tally Lambert's FP base.
Yes.
And there seem like very natural links here. Like, I kind of don't want to set up both. Have you thought about that?
We have definitely. We have definitely thought about that. We have tons of ideas. That is one of them, and other ones are I hadn't thought of, but that specific one we had is just a question of getting more. I mean, as you know, it's very hard to get funding. We've been very lucky but basically we have only one person on the Micro-Meta app side and one person on the MethodsJ2 side for now. Actually, there are two people on the MethodsJ2 side, actually. Actually, there is also Thomas, so there is more. But so yeah, we need to get more people so that we can do these things. But definitely the idea of bringing in the filters, both on the hardware side and the setting side, yeah, and also displaying. It would be great to have like some of the views that that Tally has developed for the viewer. Yeah.
Yeah. How much would it be feasible to just link to Tally's FP base? You know, not duplicate, but kind of have automatic linking to his.
Yeah, we haven't really looked into analyze like the coding side of things, how feasible it is. So I cannot tell you, but if you said that you're going to be in San Francisco, so maybe I'll be able to tell you more there.
I'd just like to make another comment. Your comment about uptake was brought up by the reviewers from Nature Methods as well, and I think you kind of have a spectrum of responses, right? Some people are like, no one's ever going to do that, why bother? And then, you know, let's make it collect every ounce of data we could possibly get, and, you know, hope people will come. But I do think that the tools is the key, and making them accessible and usable by biological scientists. And I think, you know, I was really impressed with both Joel and Caterina and all the videos they put together, because this workshop is now packaged, right? And other people could run through it on their own. So I think we're going to have a lot of work to continue to bring these tools to the different communities, and not just people who are interested in developing and programming, right, to the researchers in different fields as well. And I think, as Caterina said, I think we really have to thank Chan Zuckerberg for providing funding for this kind of thing, because as we all know, the main funding agencies want this done, but they don't want to invest in it. So, but I guess one last comment would be, I think it's really these big international networks. I've been, you know, pushing this stuff my whole career, as I know a lot of you have, and it really is the fact that we now have Bina and QuarUp and Fordia and the Chan Zuckerberg imaging scientists, that I'm seeing the companies go, wait a minute, we better get on board here. The community wants this. And I think before they were hearing lots of disparate people, you know, kind of saying, I would like this, but now it's, you know, 200 or 300 members of QuarUp want this.
Actually, 400 now.
What's that?
400 now.
Yeah. So I mean, I've been really impressed to see the impact of the networks. It's really, to me, a great case study of how important it is for us all to get together and have a big voice, you know?
Yeah, no, the companies, sorry, go ahead, Mary.
Oh, thank you. Yeah. As I said earlier, my boss wants me to implement this on the microscopes in our core facility, but I'm a little confused by a comment that Nico had made earlier. So basically, we would have to have a JSON file for every individual experiment, because the settings are going to change from experiment to experiment. Do I understand that correctly?
Yeah, so the hardware part is going to be the same for all the experiments. The settings file is, so currently, basically the idea is that there could be one per image, but if all the images are the same, then one needs to just, so like if you acquire all the different images with the same settings, you can just reuse the same settings. Yeah, but we need to, that part has to be automatized more, and we're aware of that. But yes, the idea is that you have one part that is the hardware, one part that is what settings were applied for that specific image, for that image data set.
So potentially then you could have a JSON file for each individual experiment, potentially, if the settings. Okay.
Yeah, but the good thing is that one can already now reuse a lot of the different settings files. I mean, you can reuse the files. If you have created once, you don't have to, like, for example, if you did an experiment last week, you don't have to recreate it from scratch. You can just use it again and just change a few things you need to change.
And then save it as a new file.
Yes, save it as a new file, and then associate it with that image. And
Okay.
you know, there is work to be done to make sure. So the other part of the work that we are doing, because yeah, there is tons of different aspects. One aspect is that we are working very closely with all of the open microscopy environment, next generation file formats kind of effort to make sure that this is going to be stored on the header of the files. So that once they're generated, they're going to already contain all this information. So all of this has to happen in parallel, and we're trying to keep all of this stuff going at the same time. So far, we have, thanks to the community, we have been able to do it. But you know, at least we are optimistic that we're getting there. But for the moment, there are certain aspects that have to be done manually.
And it can only do wide field microscopes right now.
For now, yes, but that is something that is really going to happen soon. To have we're actively working on implementing the confocal extension in it.
Thank you.
That's
so with wide field
on the front burner of our roadmap. Yeah.
With wide field, you mean camera based, because you have turf and spinning disk and everything.
Yeah, so for now we don't have, we are working actively on incorporating the laser scanning confocal, the spinning disk, the point detectors, all of that aspect that so far are not part of it.
Thomas?
Yeah, I wanted to ask a question actually to Nico, as we have the lucky portion of having him in the room. And it says to me that that's an interesting link, right? I mean, from a micromanager, it sounds like there would be a way to kind of export the configuration that you use for a particular image in a JSON file with the settings. I mean, you're kind of configuring it through the GUI, and then it's kind of in a certain setting. But underneath, you know the connections to all of the hardware, you know the drivers, you know the manufacturer. A lot of the information seems to be there. So I'm wondering what you think, what's your perspective on being able to make that jump from that to the JSON?
Yeah, I see your thinking. I'm afraid it is much harder than you would think. And that is because what we did in Micromanager is kind of leave this problem as far behind as we could, because it's all a problem of terminology. You know, it is like what one manufacturer calls, I don't know, a dichroic, another one calls a mirror, or, you know, like, so it's all terminology. And kind of the choice we made in Micromanager, okay, we're just not gonna, we're going to mandate only a tiny little bit, you know, every camera should snap an image and have like a very limited set of capabilities that every camera needs to have. But we know nothing about a filter wheel. In fact, I believe that an objective and a filter wheel are the same device type to Micromanager. So it cannot even tell you the difference between an objective and a filter. And that is all in the names of things that the person either, you know, what we call the device adapter imposes a name, or the person configuring the system puts in names. So it is, you know, to translate that into something like this JSON file, I wouldn't even know where to start. It would be like there would be an insane amount of conjecture would need to happen to translate these things. So in a way, I'm almost more interested in, you know, Caterina and you guys getting the whole community to settle on a common nomenclature, because then we can just, okay, we're just going to use that. And once we have that common nomenclature, you know, and we can also in Micromanager use that, then it all becomes easy, and then you can start generating these things. But it's kind of, we're going from the Wild West, and how we ever get to an organized society is like
Yeah,
maybe we
the
need
development
to
of the point. Sorry, go ahead.
Another interesting thing when we have the common terminology, and basically an ontology for microscopy, if you like, then the interesting thing would be maybe to be able to configure your experiment using the interface that Micro-Meta app has, right? Like switching the objectives and doing things like that. It seems like there could be like two-way dialogue between the different softwares in this ecosystem. So it's there's interesting things ahead. Yeah.
Mm-hmm.
Did you want to comment, Caterina?
Yeah. So the common terminology is what we're aiming for, and at least for the microscope, I think it is possible, because we have such a large community, and the whole strategy is to make sure that people can come to the table, participate to the table, and own the table, so that they can then say, okay, I had a say about this term, so it's more likely that I will use it. So 400 people that are in Quareb are primarily, for now, and that's one of the limits of Quareb, is primarily core facility managers and core facility people. So, but on the other end, this is a strength for vis-a-vis the manufacturers, because 400 people means a lot of buying power. So, so that's why I think we are able to a lot of them really, they came first and said, we really would love to do that. Like, especially like a lot of the camera manufacturers actually jumped on board very willingly, and the conversations that we are having are a lot of fun. I mean, they're very invested in, because they have this problem internally, how do you communicate even internally in the company? So, and other companies are just seeing it as happening, and they have to participate, and they're feeling like they have to participate. But clearly we get a lot of buy-in, at least formally. And so the idea of creating a shared ontology, and that's the work that we're doing with NGFF is really, with OMI NGFF, is really to transform this into a proper ontology. So yeah. So maybe the integration with Micromanager can be wait a little. It can be one of the manufacturers. When we have a more shared, a more clear model, then Micromanager can say, okay, now I am confident that this is going to be something that more than two people agree upon, and so it makes sense to invest. But still a lot of work will need to be done. So I understand that.
Are there any other questions or comments?
As far as a more abbreviated view of the metadata, definitely we are so similar. I mean, Joel showed the Excel spreadsheet to summarize things. We are definitely working on a what we call it dashboard, but basically a summarized view in which you can see the content of a file in a less, graphical manner, in a more streamlined manner. And also you can compare microscopes. For example, in a facility could be used to compare different microscopes and choose the one that is best for you to do a certain kind of experiment. As Australia Bioimaging are going along those lines, and core marketplace in the US. So anyway, we're definitely thinking of those kind of things.
Caterina, did you mention at one point there were example files of different microscopes, or that you can upload even so that others can share?
Yes, they are already available. I mean, they're not infinite. They're just a few, but I can definitely share that.
But they're not on the public site yet? Or...
They are on the public site. They are on Zenodo, but it's just files. There is no database or anything behind it. It's just files for you to download.
Definitely on the wish list, though, I think, right? To have a database of JSON files so you can pick the ones most closely matched to what you're up to.
I see a virus I see another virus and a big Olympus. Is that the files you mean?
In that link there,
there's an auto record.
You go all the way
587.
down, you can download the.
So it's only the big Olympus microscope. You only have the Olympus microscope there.
Okay, I must.
No, there's Zeiss, Leica, Nikon.
I don't see those. Where?
16 examples.
Am I in another example? Microscopy metadata JSON files produced using Micro-Meta App?
Yeah, try the link I put on the chat. Yeah, the one you have is the one that is present on the protocol IO, and is only 2 files, 1 settings and one microscope. But here the other link has 16. You see them?
No, I get 584.
That's really weird.
So this one hasn't been publicly linked to.
What do you mean? That's really weird.
I mean, how would I get to it without being here?
Oh,
I should bookmark it, or I mean, I lose this in seconds.
Because it's on.
So, email Caterina.
So what I can do is the thing I did not do, what I should have done is put it on the materials on the protocol IO. But even the protocol IO, you have to search for it. So,
yeah, I love your entrance page is the Micro-Meta app on your bymac.github.io. If you could add those links there, that would be great.
Yeah, I'll do that.
I'm just going to share my screen. We just had 2 more slides, which one of them has all the acknowledgements, so I think it's important that we see. So we just had a couple of mentions of future directions, so I think a lot of them have already come up in the discussion, but as Caterina mentioned, they're actively expanding the model now for confocal laser scanning and spinning disk microscopes, and we've had some meetings on that, and it is amazing just in the conversations we've had here too, just in terminology and how to lay out the software, and you know, as most of you would know, all the laser scanning microscopes are different. Some of them have the detectors in the scan head, and some of them have them out, and all those sorts of things. But it's in the works. Integrating the tools with the Amero database management system, which Caterina had mentioned, will have a release soon. And then we had funding from CZI as well for developing MethodsJ2 for Napari. So basically taking the same functionality that Joel showed us today, but in the Napari interface. And that's coming along quite well. And I think we should probably have a release by the fall, I would say. And hoping to get more funding to continue to develop that to be able to do more. And then the important slide here at the end, just all of the different groups that have been involved in developing these tools and disseminating them. And a big thank you to Caterina and Joel for the workshop today. And Thomas was involved in the original MethodsJ as well. So we'll leave that there, but we do have a couple more minutes if there's any last minute questions. I'm not sure the time zone for everybody, but I know it's late Friday for a lot of people. So thank you for sticking around and participating.
Yeah, the future directions that we have there are, so Judith, but anyway, let me just tell you, the future direction we have there is things that we are actually actively working on. Another big thing is the development of a database for microscopes descriptions, so that we can make it easier for people to find it, describe their microscope, and take advantage of all of this stuff. Judith.
I'm just gonna make a quick comment here. I'm a big fan of Micromanager and Micro-Meta App, and I actually have two systems that are fully documented in both Micromanager and Micro-Meta App. So Nico has kindly agreed to brainstorm with me and start exploring how we could progress in the short term. So
wow, that's great.
Let's give that a try. Sorry to put you on the spot, Nico.
Thank you.
Thanks, all of you. It's fantastic work. Fantastic work.
Thank you, everybody, for sticking around for the whole time. And see you soon.
Yeah, I guess one last comment is that all of in the instructions you received, you also have all of the videos available, and those are going to be kept on the BINA resources. So if you know anyone else who wants to hear about it or anything else, really it walks you through. Caterina's talk is there, the instructions for installing, and then this workshop itself will be recorded as well. So please, pass along those resources. And I think, as I mentioned several times, Caterina and Joel did a great job putting all those videos together. So I think it's really clear to follow, and will be able to be reused often. So
thank you.
All right, we'll finish 5 minutes early. So excellent. Thanks again, everybody.